Hi, my name is Eric Sackmann, and I'm the Associate Director of Applications Engineering here at Berkeley Lights. I'm going to talk to you today about how our new optoplasma B discovery workflow will accelerate lead molecule discovery against difficult targets. And I'll be making some forward-looking statements in this presentation. Even the best athletes in the world know that they have to take multiple shots on target in order to hit the mark. In hockey, top scorers may need to take over 100 shots on goal. As a result, these athletes aim to generate high volumes of high-quality shots in an effort to win every game. Drug discovery also requires a large number of shots on target in order to yield an effective therapeutic molecule. By some estimates, less than 5% of drug discovery campaigns successfully yield a new therapeutic. And unlike professional sports, this inefficiency is extremely problematic because each campaign can take over a decade and cost over $1 billion. Success rates for therapeutic antibody discovery against difficult targets has been especially poor, since these antibodies often need to bind cryptic epitopes or trigger complex molecular functions. To date, only 1 out of an estimated 800 human GPCRs has an approved antibody therapeutic. To close the gap, drug discovery developers need to find ways to increase the quantity, quality, and speed of lead generation. Berkeley Lights' optoplasma B discovery workflows can yield hits against difficult membrane targets like GPCRs. Genevec has successfully discovered antibodies against 3 transmembrane proteins that exemplify the common problems with difficult targets: small extracellular regions, reducing the number of possible sites to target; high sequence homology, reducing the chances of eliciting an immune response and, by extension, likely a lower frequency of antigen-specific B cells; and poor cell surface expression, making cell-based screening difficult. Across all 3 targets, they found approximately 10 times more hits than the traditional Hybridoma campaign. I'm going to describe to you how our newest optoplasma B discovery workflow expands on these capabilities to accelerate selection of lead molecules against difficult targets. Antibody discovery using the traditional Hybridoma approach requires immortalization of B cells by cell fusion, followed by a lengthy process of expansion, subcloning, and screening. The immortalization step often results in the death of greater than 90% of B cells and therefore results in a significant loss of B cell diversity. New single B cell technologies seek to access greater cell diversity by bypassing the cell fusion. However, many of these approaches provide little or no functional information. This results in significant time, labor, and expense for sequencing, cloning, re-expression, and characterizing irrelevant antibodies that do not bind the correct epitope or fail to demonstrate the desired function. Optoplasma B Discovery 4.0 maximizes diversity of antibodies through direct functional profiling of plasma B cells from immunized mice. The workflow enables users to recover thousands of hits by screening broad B cell diversity, down-select lead candidates by functional profiling, sequence and re-express over 1,000 antibodies for less than $100 per molecule, all in a week. The workflow begins with the loading of a plasma B cell sample onto Berkeley Lights' Beacon optofluidics system. The Beacon system fully automates cell sample handling, screening, cDNA synthesis, and sequence recovery. A 96-well plate containing a purified plasma B cell sample is inserted onto the Beacon. The Beacon system then automatically loads the cell sample onto OptoSelect chips. Each optoselect chip contains over 20,000 nanopens, each less than a nanoliter in volume. That's greater than 10,000 times smaller than a well in a 96 well plate. Plasma B cells are cloned into nanopens using Berkeley Lights' optoselect technology, in which cells are manipulated by light. As shown here, the light-based manipulation can be selectively performed on cells with markers for viability or other phenotypic information. Tens of thousands of plasma B cells from the target population can be cloned across multiple chips in under 4 hours. Nanopens enable rapid, precise, and highly sensitive assays. The small volumes enable secreted antibody concentrations to reach detectable levels in a matter of minutes. In the assay depicted on this slide, secreted antibodies from the plasma B cells bind antigen-coated beads and are detected by binding of secondary antibody. This results in a fluorescent halo above the pens containing antigen-specific plasma B cells. This assay is performed on tens of thousands of cells simultaneously. And the video on the slide depicts a single field of view in which hundreds of cells are screened to identify dozens of targets, dozens of hits. Antibody specificity and cross-reactivity can be assessed by performing multiple assays, either sequentially or in parallel. In this figure, we depict 3 assays running in parallel and simultaneously identifying that there are antibodies that are specific to antigen Y, antibodies that are cross-reactive to both antigen X and antigen Y, as well as antibodies that are specific to not specific to either antigen. Cell binding assays can be used to enable down selection of lead candidates against difficult membrane targets. In these assays, beads are replaced with cells that are overexpressing the target antigen on the cell surface. Antibodies specific to the cell surface target are again selected based on the fluorescent halos at the top of the pens containing antigen-specific plasma B cells. Following the single plasma B cell cloning and assays to identify the antigen-specific antibodies, OptoSeq barcoded BCR is used to automate the sequence recovery. mRNA capture beads with dual genetic and optical barcodes are loaded into specific pens, and plasma B cells are lysed to capture the mRNA. Reverse transcription is then performed on chip to generate the stable cDNA. Following the cDNA synthesis, the optical barcodes for each bead are read out by fluorescence imaging. The optical barcodes are stored in software to subsequently link each antibody sequence to its function. The barcoded beads are then automatically recovered in bulk. Over 1,000 hits can be recovered in a single 96 well plate, and additional hits can be recovered in multiple well plates if the user desires. Using this technology, we demonstrated the recovery of nearly 12,000 hits from 2 optoplasma B discovery workflows performed in a week from a single plasma B cell sample. Using a combination of fragmentation, next-gen sequencing, and bioinformatics analysis, OptoSeq barcoded BCR enables rapid and accurate sequencing of the full-length variable regions of antibody genes. The ability to reconstruct full-length antibody genes from short reads enables high-throughput sequencing of over 1,000 antibodies from a single run on a standard Illumina sequencing platform. As the graph on this slide shows, over 500 paired heavy and light chain antibody sequences were recovered from a single 96 well plate and performing a single MiSeq run. Production of lead molecules for downstream characterization typically includes costly gene synthesis and laborious bacterial cloning. Opto-BCR rapid re-expression enables production of antibodies in just 1 week to confirm function in a plate-based assay. Amplified cDNA is cloned directly into re-expression constructs, enabling expression of over 1,000 antibodies for rapid functional confirmation. In this graph, greater than 80% of the over 250 re-expressed antibodies exhibited antigen specificity in plate-based ELISA assays. The new optoplasma B discovery workflow advances from B cells to lead molecules in just a week. The workflow enables recovery of thousands of hits by screening broad B cell diversity, down selection of lead candidates by functional profiling, sequencing and re-expression of over 1,000 functionally characterized antibodies. By increasing the quantity, quality, and speed of generating shots on goal, Berkeley Lights' optoplasma B discovery workflow will enable drug developers to discover lead candidates against difficult targets. Thank you, and please reach out if you're interested in learning more.